lapel.
Welcome to the Deep Dive. We're here to take that stack of sources, pull out the core insights, and basically give you that shortcut to being really well informed. And today, we've got a really vital mission, actually tailored specifically for you if you're prepping for PhD quals in pharmaceutical sciences. We're diving into biosynthesis and drug metabolism.
That's right.
We've got this key chapter, and our plan is to break it down, make it stick, and explain it so clearly you could, well, turn around and give a lecture on it yourself.
Yeah.
Think of this as your focused study session.
Exactly. We're going on quite a journey today, starting right from the origins of life, really, up to the incredibly complex cellular machines that protect us. And that process that the medicines we rely on, we'll look at how your body handles, well, everything from ancient toxins to modern pharmaceuticals.
And even how our own systems can sometimes be fooled, right?
Yes, precisely. It gets quite intricate.
Okay, so let's get started. We're diving deep into human drug metabolism, kicking off with its, well, ancient biological roots. First up, the very dawn of these enzymes. When we think about early life, it's kind of mind-blowing biologically speaking. Before bacteria, there was nothing. And early Earth wasn't exactly welcoming. Imagine outer space, but, on a planet just forming. Extreme heat, cold, radiation.
Corrosive chemicals, no oxygen. It was incredibly hostile.
So life needed some serious resilience just to get a foothold.
It really highlights an almost unbelievable adaptability, but bacteria. Their growth rates, their generation times are just phenomenal. That let them evolve enzyme systems incredibly fast, allow them to survive, even thrive in all sorts of harsh news.
That's why people even talk about bacteria maybe under the surface of Mars, right? That adaptability.
It speaks volumes, yes. Their evolutionary success is just staggering.
So, okay, life evolves. We get eukaryotes, more complex cells. And a key feature is this watery cytoplasm inside a fatty lipophilic membrane. Now, that membrane is tough. You mentioned tardigrades surviving in space.
Right, incredibly robust.
But here's the twist. It's not a barrier to other fatty lipophilic molecules.
And that's the crux of the problem. Really lipophilic stuff can diffuse right into the membrane, get trapped, mess things up. Slightly less lipophilic molecules, they can pass right through into the cell.
Okay.
So early life needed more than just basic housekeeping enzymes. It urgently needed biotransforming enzymes. To detoxify internal reactive stuff, yes, but also invading molecule. Waste from other bacteria, maybe, or aromatic hydrocarbons from fires. Incomplete combustion.
And if they weren't dealt with?
They just build up, accumulate. They can derange enzyme systems, damage cell structures. Big problems.
And this isn't just ancient history. Organisms today that can't handle these lipophilic hydrocarbons, they accumulate them, sometimes to toxic levels.
There's that striking marine example, isn't there? The sponge, hymeni acid and prolific.
Right, versus the Pacific oyster. The sponge had 17 times more benzopyrene.
Exactly. It suggests its biotransformation capacity just couldn't keep up, a failure to clear it.
Which brings us right up to today. Our whole society relies on petrochemicals. So these lipophilic hydrocarbons are everywhere. Air, food, water.
It's a huge exposure. And think about dioxin. PCDD, that's like the worst case scenario. Carcinogenic, teratogenic, causes birth defects, potent endocrine disruptor.
And the half-life.
About
decades in humans.
Can be, yes. The body mounts a response, tries to deal with it, but it really struggles to eliminate it quickly. It just persists.
So how do we cope? How do almost all living things manage this constant chemical onslaught?
The fundamental answer lies in a truly ancient and widespread enzyme system. The cytochrome P450 monoxygenases, CYPs, P450s.
C450s, right. Named for how they absorb UV light.
At 450 nanometers, yes, when they're reduced and bound to carbon monoxide. Very specific signature. Their origins are incredibly old, probably evolved way back in Earth's early CO2 atmosphere, maybe for reducing things initially.
But now they mainly do oxidation.
That's their main job now, yes. Oxidation. And what's amazing is, it's not just one enzyme, it's a huge gene family. Super family, really. Think of it like a set of adjustable spanners or wrenches in a toolkit.
Okay. Each one's slightly different?
Each one subtly adapted to tackle, to dismantle particular chemical structures. It's incredibly versatile.
That's a great analogy. So these CYPs, they represent billions of years of nature's R&D.
2 to 3 billion years, yeah. That's why they're so successful.
Meaning, even with thousands of new man-made chemicals appearing each year.
The vast majority, probably metabolized by at least one CYP form. The system has incredible breadth. It's a testament to that long evolution.
Okay, so ancient defense systems are in place. Fast forward.
Yep.
Organisms get bigger, multicellular. Now you need communication, coordination. How does that work?
Exactly, a whole new set of challenges. Advanced organisms basically develop two main communication lines. Direct electrical nerve impulses. Think sympathetic nervous system hitting the adrenal gland.
Yeah.
Instant. Or chemical instructions. Hormones, mostly. Often slower, but more widespread, perhaps more profound effects.
So if you're going to use a chemical as a signal, it needs certain properties to be reliable, right?
Yeah.
What makes a good chemical messenger?
Good question. There are essentially four key features. First, stability. It can't just change shape randomly. Needs to dock reliably with its receptor.
Makes sense.
Second, resistance. It needs to resist reacting with other random chemicals or enzymes in the body. Has to deliver its message intact.
Okay. Stability, resistance. What else?
Third, manufacturability. The body needs to make it easily in large amounts using available building blocks.
Huh. You said the pharmaceutical industry thinks the same way.
Very similar criteria for drug design, yes. Availability of starting materials, ease of synthesis. It's a parallel process.
Fascinating. Okay, and the fourth feature?
Controllability. This is crucial. The signal can't stay on forever. That would be wasteful, potentially harmful. So you need balanced production and elimination. Fine-tuning the concentration, the duration of the signal.
But isn't there a contradiction there? Stable enough to work, but easy enough to get rid of.
Absolutely. It's an inherent tension. Stability versus disposability.
Yeah.
And both making it and breaking it down costs energy. It's a biological balancing act.
Right. And for those big, slow changes, like reaching sexual maturity, nerve signals aren't enough. You need chemicals, right? Controlling genes across millions of cells, maybe for years.
Exactly. Direct neural control isn't suited for that kind of sustained programmed change. And this is where lipophilic hydrocarbon chemicals really shine as signals. They offer stability, they're made from plentiful precursors, and their solubility can be tweaked.
Tweaked how? And where did the CYPs come back in?
Well, this is where it connects beautifully. Those CYPs we inherited from bacteria, they've evolved to radically alter the shape, solubility, stability of these aromatic molecules. They essentially allow the custom building of stable signaling molecules, often using cholesterol as a basic scaffold.
So, like, cholesterol itself is super fatty, gets stuck in membranes.
Exactly. Very lipid soluble, practically zero water solubility. Trapped. But steroid hormones, built from cholesterol. CYPs add just enough hydrophilic groups, water-loving groups, to make them less lipophilic.
So they can travel.
They can travel, yeah. Bound to carrier proteins in the circulation, then slip into cells to reach their nuclear receptors. But they're not so water-soluble, they just wash straight out.
There's a sweet spot for solubility. You mentioned ostradiol versus benzene.
Right. Estradiol 17 beta is about twice as water-soluble as benzene, but still way, way less soluble than something like vitamin C, about 92-fold less. It's mobile, but persistent enough.
And then, when the signal's job is done,
other CYPs step in again. They add more hydrophilic groups, increasing water solubility further, tagging them for excretion by the kidneys. Like converting benzene to phenol makes it 46 times more water soluble, ready for removal.
Okay, that system makes sense, but it also seems vulnerable. What about things that look like signals but aren't? False signals.
Now you're hitting on a major real-world problem. Endocrine disruption.
EDCs. Endocrine disrupting chemicals.
Exactly. It turns out our estrogen receptors, for example, are, well, somewhat promiscuous. They'll bind to quite a wide range of chemicals, natural and synthetic. Things like polycyclic aromatic hydrocarbons from burning stuff, certain plant toxins, man-made chemicals, BPA, phthalates from plastics, parabens in cosmetics.
Because they have similar shapes, similar aromatic rings.
Often, yes. They mimic the natural hormone structure well enough to bind and activate the receptor. And a big worry is their persistence. Many have long half-lives. They hang around.
And this isn't new. You mentioned an arms race.
It's ancient. Plants evolved toxins to deter animals. Animals evolved defenses. That big expansion of the CYP gene family, maybe 300, 400 million years ago when animals came onto land, that likely reflects this intense evolutionary pressure, dealing with new plant chemicals.
Oh, CYP2D6 preferring plant alkaloids, and some groups having more copies of that gene.
Precisely. It suggests an adaptive response to dietary pressures over generations, a genetic defense.
But there's a dark side. Our own defenses can backfire.
Unfortunately, yes. Sometimes, our CYPs can interact with toxins like mycotoxins in a way that actually increases their toxicity. But here's the really counterintuitive part, especially regarding EDCs. Sometimes, our biotransformation systems can inadvertently promote the disruptive effects.
No.
Take benzopyrene again. The parent molecule is super lipophilic, mostly trapped in membranes, limited access to receptors inside the cell, but when our CYPs metabolize it, they create hydroxylated metabolites like the 7,8-diol epoxide. These are more water-soluble.
Ah, so they can get to the estrogen receptors more easily.
Exactly. The body's attempt to detoxify and eliminate it actually creates a metabolite with better access to the target site, potentially making it a more effective endocrine disruptor.
Wow, okay, that's deeply concerning. Our own system trying to help makes it worse.
It highlights the complexity, and it's not just synthetics. Plant phytoestrogens saw palmetto, fenugreek in Act 2. They partially fool or modulate our systems.
And you mentioned microRNAs from food? Influencing our genes.
It's an emerging area, yeah. miRNAs that can survive digestion get absorbed and potentially influence gene expression, including our biotransformation systems. The gut-gene communication axis is getting really interesting.
Okay, so we understand the why and the evolutionary context. Let's get practical. Where does this biotransformation actually happen? The workhorses.
Right. While CYPs are basically everywhere doing essential jobs like making steroids, not just clearing drugs, the highest overall capacity is definitely in the liver and the gut.
Why them specifically?
It makes sense physiologically. They're the first line of defense, processing everything coming in from the gut via absorption and things circulating in the blood.
And the liver has that unique circulation you mentioned.
The dual blood supply, yes. It gets oxygenated blood via the hepatic artery like other organs. But crucially, it also gets blood directly from the entire GI tract via the hepatic portal vein.
So, food, drugs, toxins, recycled bile salts, everything absorbed goes straight to the liver first.
Pretty much. The liver cells, the hepatocytes, are bathed in this mix from both supplies. It ensures xenobiotics get presented for biotransformation immediately. First pass metabolism.
And once processed in the hepatocyte, where do things go?
Two main exit routes. Either back into the sinusoidal blood, which eventually flows into the hepatic vein, systemic circulation, and towards the kidneys, or into the bile via tiny channels called canaliculi leading to the bile duct, and then back into the gut for elimination in feces.
Is there a rule for which way things go, blood or bile?
There's a general tendency. Larger molecules, especially anions, above a certain molecular weight, roughly 475 daltons. Daltons, as often cited, tend to be preferentially shunted into bile. It's related to molecular volume and relies heavily on those ABC cassette transporters we'll discuss.
Okay. Liver and gut are the big players. What about other organs? Lungs, kidneys, skin?
They contribute, but less to overall drug clearance. Their significance is often local. For instance, CYPs in the lung can activate inhaled compounds into reactive species, potentially contributing to lung cancer. So local toxicity is a key issue there.
And the brain. You mentioned that too.
Lower overall CYP levels, but very specific functions. We used to think mainly neural steroid regulation. Now we know they're crucial for protecting the brain and astrocytes at the blood-brain barrier, clearing xenobiotics, and they can really impact drug therapy, epilepsy, for example. Sometimes treatment fails because brain CYPs clear the drug too fast locally, or they form metabolites that are actually epileptogenic.
Right, so local metabolism can really change the game. Now, getting into the cells, it's not just simple diffusion for everything, is it?
No, absolutely not. Entry isn't guaranteed. As we said, very lipophilic drugs get stuck in membranes. Charged drugs or amphipathic ones with mixed properties diffuse poorly.
So you need help getting across the membrane.
You need transport systems. Membrane transporters are critical, found all over, but especially key in the gut lining and on hepatocyte membranes for absorption and processing.
And these fall into families.
Two main superfamilies. First, the influx or uptake transporters. Mostly silate carriers, or SLCs. These help things get into cells. They don't directly burn ATP energy. Found in liver, gut, brain, kidney, placenta, vital locations. Huge family, over 400 proteins from 52 gene families in humans.
Okay, influx, SLCs. Which ones are most important for drugs?
Several key subfamilies. One major group is the SLCOs. These make organic anion transporting polypeptides, OATPs. Another is SLC22A, coding for OATs, organic anion transporters, and OCTs, organic cation transporters.
Let's take OATPs. How do they work? You mentioned a revolving door.
That's a good way to think about it. They're sodium independent, they facilitate diffusion. For every molecule they bring in, like an amphipathic drug, they push an intracellular anion out, like glutathione or bicarbonate, to maintain charge balance.
And lots of common drugs use these. Statins.
Many hydrophilic drugs, yes, statins are a prime example. And this is clinically huge. If you inhibit an OATP, like OATP1B1 in the liver, using another drug, say gemfibrozil or rifampicin, you can block the uptake of atorvastatin into the liver. That blockage can raise blood levels of atorvastatin far more dramatically than inhibiting its metabolism would. It's a major site of drug interactions.
Wow, okay. So transporter inhibition can be even more impactful than metabolic inhibition sometimes. What about the OATs and OCTs?
OATs like OAT1 and OAT3, mainly kidney-based. They pull small molecule drugs and waste products out of the blood into the kidney tubules for excretion, very important for renal clearance. Then there's the SLC10A family, bile salt transporters. NTCP, ASBT, they recycle bile salts that enter hepatic circulation.
And they move drugs too.
They can, yes. NTCP is important for hepatic uptake of drugs like rosuvastatin. ASBT is in the kidney ileum. Both are crucial, and like OATPs, they can be inhibited by drugs, messing up bile salt flow and drug handling.
Okay, that covers influx via SLCs. What's the other major superfamily?
The efflux transporters, pumping stuff out. These are mainly the ATP binding cassette, or ABC, transporters. They use ATP energy to actively pump molecules out, often working against a concentration gradient.
These are the ones involved in cancer drug resistance.
Exactly. They were heavily studied for pumping chemo drugs out of cancer cells. The genes look a lot like bacterial antibiotic resistance genes, MDR, multi-drug resistance. In the liver, they're vital for moving those polar metabolites, the results of phase 1 and 2 metabolism, out of the hepatocyte, either into the blood towards the kidney or into the bile.
Okay, let's pull this together. We have lipophilic drugs, hard to absorb well, but also hard to get rid of. They hang around, get stuck places.
Right. Filtered by the kidney, maybe, but then reabsorbed. Trapped in fat for years.
What are the dangers of them just hanging around?
Well, trapped in membranes, they affect fluidity, function. They can trigger unwanted, irreversible hormonal effects if they mimic hormones, direct damage to proteins, DNA, and just prolonged drug effects, which might not be wanted. So the whole system has this dual challenge. Build necessary molecules like steroids and clear them, plus defend against all these external xenobiotics.
It detects them and organizes their removal.
Essentially, yes. The primary task is taking something very oil-soluble and making it highly water-soluble.
Like washing greasy dishes, you need detergent.
Exactly. The detergent here is structural modification, changing the molecule's properties so it dissolves in water and the kidneys can flush it out.
Now, traditionally we talk about phases, phase 1, 2, 3. Can you quickly recap those?
Sure, the classic view is phase on is mainly oxidation by CYPs, but also other things like reductions, hydrolyses, basically adding or exposing a functional group. Phase 2 is conjugation, sticking a water-soluble endogenous molecule, a sugar, a sulfate, an amino acid onto that functional group. And phase 3 is efflux, those ABC transporters pumping the conjugated water-soluble product out of the cell.
But you said this terminology is problematic, misleading.
It can be, yes, especially if you're aiming for deep understanding, like for your PhD exam. First, phase I lumps together very different types of reactions. More importantly, it implies a strict sequence, I, then 2, then 3, and that's just not always true.
How so?
Well, a phase 3 efflux pump might kick the original parent drug straight back out of a cell as soon as it gets in, before any phase I or 2 happens. Or a drug might only undergo phase 2 conjugation with no phase I needed. Or only phase I oxidation before excretion. The sequence isn't rigid.
So what's a better way to think about it? A more precise classification?
It's better to talk about functionalization reactions and conjugation reactions.
Okay, define those again.
Functionalization. Adding or unmasking a polar handle. An AAH NH2 bi-COH group makes it a bit more water-soluble or prepares it for the next step. Conjugation. Covalently attaching a big water-soluble endogenous molecule like glucuronic acid or sulfate onto that handle.
Less soluble, so they could crystallize out.
Precisely. Precipitate in the kidney tubules cause kidney damage, so conjugation isn't inherently safe.
That's a critical point. Okay, usually metabolism reduces or kills the drug's activity, but not always. You mentioned metabolites being more potent.
Yes, this is where it gets really interesting pharmacologically. Some metabolites can retain significant activity or even be more potent than the parent drug.
Can you give that example again?
The classic one is morphine-6-glucuronide, M6G. It's a phase 2 metabolite of morphine.
And it's stronger.
Significantly, maybe 100-fold more potent as an analgesic than morphine itself at the opioid receptor.
Wow, 100-fold from adding a sugar molecule.
It's quite remarkable. However, remember the flip side. That increased polarity, that big sugar group, usually makes it a prime target for rapid efflux by transporters. So while it might be...
Pharma